friends, this is Prashant Katiyar. I am the faculty of the Life Science Academy. This is the premier institute of GATE and CSI NET exam preparation, which is located in Prayagraj. So in this video we discuss about glyoxylate cycle. Glyoxylate cycle. First, you have to know where this cycle is occurring, and this cycle occur in mostly in plants and in case of invertebrates, but this cycle not occur in invertebrates. So first point is this cycle occur in invertebrates and plants. Okay, invertebrates, bacteria, for example E. coli, some Erythrobacter, some Bacillus species like Bacillus paracus, where this cycle is usually occur. But in case of plant, when plant stores excess fat or fatty acid in the form of lipid as triacylglycerol, they are oil droplets which are stored in adipose tissues. Okay, so when this fatty acid is synthesized, when the fatty acid is synthesized in excess amount, this fatty acid is stored as triacylglycerol in adipose tissues. Okay, here we call these tissues as lipid body. What is lipid body? So we picturize this lipid body as in this way. Suppose this is the lipid body where this triacylglycerol is stored. Okay, so when this plant, in case of plant, when the seed is germinating, okay, then this excess of fat is break down. Okay, this triacylglycerol is break down in fatty acid. Okay, and this fatty acid when this fatty acid is liberated and comes into the cell organelle, which is formed at the time of seed germination. So that is called glyoxysomes, where this cycle occur. Okay, this cycle, glyoxylate cycle, occur in glyoxylate. This is the cell organelle where glyoxylate cycle is occurring in case of plant and in case of bacteria. Okay, so when this braked fatty acid comes in this glyoxysomes, it again oxidizes and comes into this form acetyl coenzyme A, that is the 2-carbon compound, and this acetyl coenzyme A combined with oxaloacetate. So this fatty acid is break down in smaller unit, that is called acetyl coenzyme A, that is 2 carbon compound. Okay, when this acetyl coenzyme A, now because as fatty acid is okay, so this fatty acid is oxidized into in glyoxysomes compartment. This is the glyoxysome compartment where fatty acid is oxidized into acetyl coenzyme A, and in this compartment oxaloacetate is already present, that is the 4 carbon organic compound, and this 4 carbon oxaloacetate combines with this acetyl coenzyme A to form a new compound that is called citrate. Okay, then this citrate is again generate a new product that is isocitrate. Okay, now this isocitrate act as a bridging element between glyoxylate and next upcoming cycle, that is the citric acid cycle. So this is the major bridging element which now this isocitrate is again generated a new product in the presence of citrate, in the presence of lyase. This is enzyme which cleaves this isocitrate into 2 compounds. One is succinate and another one is glyoxylate. Now this 6-succinate diffuses from this cell organelle glyoxysomes. This is the boundary, outlet boundary of this glyoxysomes. Okay, now this succinate diffuses from this outer outlet boundary of the glyoxysomes and comes into the cytosol, where it again come into the mitochondria, where TCA cycle occur, or you can say citric acid cycle. Okay, now this succinate comes into the mitochondria. Suppose this succinate comes into the mitochondria. Okay, this is the mitochondrial compartment where TCA cycle or citric acid cycle occur. Okay, now this, why it is comes into the succinate. Okay, why this succinate comes in this mitochondria compartment? To generate malate. To generate malate. See, this malate again moves outside from this mitochondrial membrane, because this malate is permeable to this membrane, because some transporter proteins which is specific for this compound is present here in the outer membrane. So it passes from this transporter protein and again come into the cytosolic compartment, where this malate again converted into oxaloacetate, and this oxaloacetate generated PEP, that is called phosphoenol pyruvate. Phosphoenol pyruvate in the presence of pyruvate carboxykinase. This is the enzyme which convert oxaloacetate into 3 carbon compound with the liberation of 1 moles of CO2 in the presence of pyruvate carboxykinase. This PEP, generation of this compound indicates straightforwardly that this compound is entering into the gluconeogenesis process, where this is the non-carbohydrate part. This is not a carbohydrate compound. This is the organic compound which generated ultimately hexose, or you can say glucose. Now this glucose is ultimately converted into sucrose, where in this form glucose is transported in roots and shoots and other organs of the plant. In this form, in this form of sugar, usually sugar is transported to the roots and shoots of the plant. So this is the all about glyoxylate cycle. One thing I want to clear it, the generated glyoxylate cycle. What about this? This glyoxylate again fuses with 1 mole of acetyl coenzyme A and generate malate, and malate generate oxaloate acetate. So until whole seeds containing lipid, and suppose seeds having 25 milligrams of lipid, suppose, so 25 milligrams whole lipid, if converted into this glucose, until till that stage, this cycle is going on, okay, in this way. So what are the major key regulators for these cycles? One enzyme is this isocitrate lyase, which is a actual regulatory element of this glyoxylate cycle, which cleave this compound isocitrate into succinate and glyoxylate. So this is the major regulatory element. And another one is when the other intermediates of citric acid. Okay, so intermediates of TCA cycle and glycolytic intermediates, as well as AMP and ADP level, intermediates of citric acid cycle like fumarate, malate, oxaloacetate, all the citric acid cycle, citrate. So when this citric acid level, intermediates of citric acid cycle and AMP ADP level is these all are accumulated, then it clearly indicates the energy depletion stage. So at this stage, this glyoxylate cycle usually occurs, okay. So reversibly, if it is not accumulated, if these factors are not, these intermediates are not accumulated, then it clearly energy deplete, that is not a energy depletion state. It means cell having enough amount of energy. So plants, or in case of plants or in case of invertebrates, there is no need to run this cycle, okay. So this is the all about glyoxylate cycle. So main point is, students, you have to remember this thing. The key regulatory enzyme of this cycle is isocitrate lyase, which having a function of cleavage of what? Isocitrate. This is the bridging element of element between glyoxylates and TCA cycle. Okay, and then another point is these factors which indicates the energy depletion stage. At this stage, glyoxylate cycle is usually runs. Okay, so in what form sugar is transported into the root and shoots? It transported in the form of sucrose. So these are the crucial and important points. In addition, how many, if competitive exams like GATE and CSI are not net, um, some students faces uh, regular uh, uh, so the main precursor of this cycle is acetyl coenzyme A. If 2 moles of acetyl coenzyme is producing from this fatty acid, then how many moles of succinate is producing? There is only 1 mole of succinate is producing. Okay, so this is the question which arises usually in GATE and CSIR NET exams. Okay, so this is the main points of these cycles. You have to focus on these points. Okay, thank you students. If my video is you are getting all the points, then subscribe this video. Okay, thank you.